The overall goal of this procedure is to fabricate a polyelectrolyte complexation shell, a carrier that can entrap and regulate the release of a wide range of growth factors with the heparin binding domain.
These methods can answer key questions in orthopedic tissue engineering field through efficient entrapment and delivery dedicated growth factors. The main advantages of this technique is it does not require usage of organic solvent, which is known to affect the bioactivities of growth factors.
Dissolve 200 milligrams of non-irradiated sodium alginate or 400 milligrams of 8 megarad irradiated sodium alginate in 10 milliliters of double distilled water and shake for 1 hour. Store the alginate solution at 4 degrees Celsius overnight. Filter the alginate solution with a sterile 0.2 micron syringe filter before alginate microbead fabrication. Disinfect the electrostatic bead generator and syringe pump with 70% ethanol and place them in a Class II biological safety cabinet. Then, place a glass basin with a magnetic stir bar inside the bead generator. Set the arm electrode of the bead generator 9 centimeters above the basin. Connect the electrode cable of the bead generator to the knurled screw 2 of the arm electrode and pour 80 milliliters of